Good morning and welcome to Conversations with JoAnne Stubbe. I've been asked to introduce Professor Stubbe. My name is Stuart Licht. I was a graduate student in Professor Stubbe's lab, and I was to be...
You used to call me?
Yes, always. Always. Still. And I was surprised, to be honest. I've been asked to be somewhat expansive in my introduction, so it's not just going to be, here's JoAnne. So you'll have to sit tight for a few minutes. I was surprised, frankly, to hear that JoAnne had agreed to do this. I probably, in conversations with JoAnne, often tend to come in the form of an argument. And I would have thought that she would have said something like, what's interesting about a scientist is science. So if you want to have a conversation about what's interesting about me, I'll give a seminar and you'll ask questions and we'll have a conversation about science. And that's what's interesting about a scientist. But then I thought, well, this is, and like usually, JoAnne made a good point. She didn't actually make it, but she would have made it. And I thought, well, but that's not quite true. JoAnne has had a very interesting career. She's, told me stories, and certainly graduate students in the lab, about mentors she's had that have been valuable to her, and situations where she had to be, more independent than she might have felt prepared for at a given time, or the path she took to MIT is unusual. She at one point told me that she'd been an assistant professor for 14 years. Maybe she'll talk about that. And I thought, well, those things, she's got a lot of interesting stories about that. And then I thought JoAnne would probably say, but that doesn't have anything to do with science. That's about the job of being a scientist, but that's not interesting. That doesn't have anything to do with science. And finally, I came to the conclusion that, at least, and I'm not sure JoAnne can agree or disagree about the value of this, but for me, what has been interesting over the years as a student and as a colleague about observing the way that JoAnne thinks about science and does science is the process that she goes through. So something that's not as, or that's more unusual than it should be is, of course, JoAnne, like pretty much everybody at MIT, is very focused on a question. Let's find an interesting question. Let's ask an interesting question. But from there, she proceeds really from the bottom up. She'll ask, what buffer is in your reaction mixture? Every salt, every component. Experimental details are paramount. And only from understanding these completely will she concede that at some point a point has been proven. It always struck me as a graduate student that JoAnne, of course, as those of you who know her are familiar with, is forceful in her style of debate. And you would have to always defend what you did in a group meeting, when you talked with her. But you would always come under the most intense scrutiny when you had a result that looked like it was something that she actually believed was true. And this has been a lesson that always stuck with me, that she was most stringent with the beliefs that she cherished the most. And this was a forceful demonstration of what I think is extremely interesting about JoAnne, is that over and above the science as a career, science as a job, science as a way to get fame and fortune, or to the extent that one does, JoAnne is so dedicated and passionate about science that she has come up with a process that allows her, to serve her ideals of science. And, and then that sounds high-flown, but it works itself out in practice in a very concrete way. And just to wrap up, you see it, or at least I think anybody who's worked in the lab sees it on a day-to-day basis. I remember distinctly having an experiment in the scintillation counter, and this was tritium, the low-energy emitter, so these were 5 minutes counts, and watching JoAnne watch the counts come off the scint counter, and thinking, I thought to myself, this is not an efficient use of her time. But, then I thought, to what extent is any of this an efficient use of anybody's time? And really, I think that shows what JoAnne is about. I've often thought that if everybody loved something as much as JoAnne loves science, the world would be a pretty amazing place. So without taking any more time from conversations with JoAnne Stubbe, here's JoAnne.
That's it. So I didn't prepare anything, but I'm JoAnne, and this is my dog. My dog's name is McEnzyme, okay, and that's because I'm an enzymologist, and so we call her Zyme.
What breed is that?
She's a Cairn Terrier, and she loves to go to group meetings. She's the comic relief in my group meetings because she loves to sit and watch the laser pointer. And she jumps on you, and then she jumps on the black. Every time somebody's talking about an exciting result, she jumps up on the blackboard and wags her tail, so you know you have an exciting result. So anyhow, I think because Linda thought that I was going to be pretty boring, she asked me to bring my dog as a way of having more comic relief. So I don't know what these things are about. Amanda sort of twisted my arm to come do this. So I guess I could tell you a little bit about my history, and if you want to ask questions, you should interrupt. I'm in my early 60s, and I think that makes a comment on my scientific career, as you'll see, which has been very different from most young people's scientific careers. I was born in the Midwest, Champaign-Urbana, the cornfields. My father used to teach at the University of Illinois, and I don't remember that at all, but I stayed there for 2 years, and then he moved to the Worcester area, and so that's where I've spent the whole rest of my life. So I'm a confirmed New Englander, and I come from Worcester, Mass, if you can't tell that I'm a confirmed New Englander, and I've had many opportunities to move back and forth, but I think my heart is actually in New England for whatever reason. So anyhow, I guess people want to know how you got interested in science. I was interested in nature. My grandfather, everybody in my family worked. My mother worked, she was a teacher. My father worked, my grandmother worked. My grandfather didn't do a day's work in his life. He liked to hunt and fish. And so some of my fondest memories were actually walking in the woods with him, looking at wildflowers and looking at birds. And so at a very early age, I was really interested in that aspect of biology, not knowing the names really, but I thought flowers were beautiful and I thought birds were cool. And so I had a nature club. And what I used to do is enlist all the people in the neighborhood when we'd have a project in school to figure out who could get the most wildflowers and name the most wildflowers. I would enlist all the people to go get 5 wildflowers, and I always won all the contests because I always had the most wildflowers. So anyhow, I think that was my first introduction to nature. I mean, I love being outside. I love trout fishing with my grandfather. I love walking in the woods. Other than that, I really wasn't all that excited about science till I got to high school, where I had a fantastic chemistry teacher, Mr. McHugh. I remember his name. And what I liked about chemistry, in contrast to most males, was the colors and the crystals. I hated the explosions. And in fact, on numerous occasions when I was in undergraduate, I almost quit. Because, back in those days, you used to use Bunsen burners, and you were washing your hands in benzene, which is a carcinogen. You were dumping ether down the drain, and then all of a sudden your Bunsen burner would get caught in the flame, and the whole trough where the solvent was going down the drain would be lit up. So these are really different days. That almost got me out of science completely because I was so scared of the explosions and the bad parts, which I think get a lot of people excited. So my enthusiasm, which I still really like, is seeing something crystallize, mostly inorganic salts that are colored. Seeing things crystallize and seeing changes in colors, and over the years I've worked on vitamins like vitamin B12, which is nature's most beautiful cofactor. If you work on it, you're not supposed to shine a light on it because it's light sensitive, but it turns from pink to orange. And you can tell actually by looking at the color, if you steal a peek, exactly what the oxidation state of the metal is. And so those are the kinds of things that got me excited. And I never had any biology in high school. Never had any biology in college. Never had any biology till I had to teach it when I was an assistant professor at Williams many, many years ago. And so that's teaching what got me excited about what I do today. In contrast to most people who sort of start out and they get excited in their PhD program and then they go on and do another postdoc and get more excited. My sort of real enthusiasm didn't come till I took my first academic job. So I was always interested in science, though, subsequent to my interaction with this chemistry guy at Classical High School, because my father was a faculty member. He got me a job working in somebody's lab, even though I knew no organic chemistry. And so starting, I think it was when I was a senior in high school, and then my freshman, sophomore, and junior years in college, the university had an NSF fellowship program in the summer, and I used to work there. And this guy, his name was Ed Trachtenberg, really had a strong influence on my career. Taught me organic chemistry when I was in high school, basically. So I had never had anything. I didn't even know what carbon was, and I was in there. I just love working in the lab. And as Stuart says, I love the details. That's what, I always regret that I'm a faculty member now because all I do is BS all day long. I write hundreds of letters of recommendation. I do all this stuff I hate. And you guys are the ones that get to do all the exciting things and see the changes that make a difference in what happens. And so he says, I'm looking at the scintillation count. You're damn right, except I wouldn't have left it there for 5 minutes. I would have turned it to 30-second counts, go right to the area, and saw if I saw a peak in the region where I thought something was going to happen, whereas you guys turn it to 20 minutes and go home overnight. What can I say? Still, that's what drives me.
I knew I'd have to tell you what the standard deviation was on the counts. And
No. I'm smart enough to realize that requires careful quantitative analysis. So anyhow, so I had all of that experience. When I was an undergraduate, I didn't work in the laboratory till my senior year, although back in those days, you typically took 5 chemistry courses a semester, 4 or 5. You took a lot more courses than you take now, and every course had a lab associated with it. In contrast now, we have to be out of the lab by 5 o'clock or you call the dean. We used to be in the lab till 8 and 9 at night, and I love doing things like getting a bunch of unknowns and figuring out how to separate the unknowns, and the discovery that all of a sudden, man, I know what I have. These are the 5 compounds I have in this mixture. I think got me really excited about science. But in addition to chemistry, which I decided to major in, I was also very interested in math. I was a math major up until through my junior year. And I was also interested in art history. And my interest in art history has still carried over. For example, when I first got here, I taught a course in chemistry and art. And when I first went to graduate school, what I really wanted to study was, I went to work for a photochemist and studied the effects of light on painting. So I could work in restoration for someplace like the Fine Arts Museum. But the photochemist that I wanted to work for didn't take women into his group back in those days. So that was the end of my photochemistry experience. So anyhow, as an undergraduate, I think my greatest passion was not chemistry. My greatest passion was tennis. And my second greatest passion was squash. And then maybe chemistry came after that. I mean, I remember Ed Thornton, who I did undergraduate research with, came and said, R.B. Woodward. How many of you know who R.B. Woodward is? Okay, so there's a few of you. Okay, I mean, like he was the great Greatest, yeah, he was the god. I mean, and still is. I mean, if you look back what he's done, most people don't know who he is, and that's a comment on scientist contributions. Anyhow, nobody knew nobody knows who R.B. Woodward was. I knew who he was, but I had a tennis match, and so that was it. I said, sorry, I can't go here. I got to go play tennis.
Where were you in college?
So I went to, so I went to the University of Pennsylvania. My parents had no money. We really, literally had no money. I started working when I was 12 to save for college and worked all the way through high school. I decided where to go to college because in those days they had tuition exchange plans, and so I only applied to places where I got tuition free. And so all my brothers and sisters and I, luckily, were okay. We got into good schools. And so, yeah, I thought Penn was, I got an outstanding education there. I mean, when I went to graduate school, I had no problems at all because my education was so good. But going back to what we took many, many chemistry courses compared to what you guys take now. And every single course had a lab associated with it. So every afternoon throughout sophomore, junior, and senior years, you were in the laboratory all 5 days. I mean, that's just the way it was, and sometimes at night. So that part, and back in those days, did they do things like UROPS? Not really, but I did a senior thesis project with Ed Thornton. And both Ed Thornton and Ed Trachtenberg, who is this faculty member I worked with in the summer, were physical organic chemists. And so physical organic chemist is somebody that studies the mechanism of how some small molecule gets converted to some small product. And that really was my underpinning. I was excited about mechanism, and I still use that underpinning in everything I do today. So when it was time for me to apply to go to graduate school, I had no idea. I mean, you know, my what do you call it, advisor, okay? The way you communicated with advisors back in these days, they had a clipboard. You put what you wanted to take, they signed, and you put it there, they signed it, you picked it up. You never saw your advisor. I don't even remember who my advisor's name was. Anyhow, so to apply to graduate school, I had no idea where to apply. And so I applied to places on the East Coast, not even looking at what the quality of the departments were, because I had no idea how to look at that. And of course, nobody told you anything about that. And the guy that I had done, Ed Thornton, who I had done research with, who went to MIT, convinced me that I should go to the best chemistry department in the country, which he said was Berkeley back in those days, but I didn't know from a hole in the wall. And so that's where I applied there, I got in, and he convinced me that I should go there. And I think if I hadn't gone there, I wouldn't be here, okay? Because the name does make a difference, okay? And so that was completely by accident that I happened to go to Berkeley to graduate school. And I remember, you know, I'd never flown anywhere, I'd never been anywhere. In fact, the first time I went to a restaurant was when I was undergraduate at Penn, where I had some very rich, preppy roommates whose parents took me out to dinner. I'd never been to a restaurant before that. And so I had to fly to California by myself, you know, into this place. And then when you flew to San Francisco, you'd take a helicopter ride from the San Francisco airport to the Berkeley airport, which was really scary. I still get scared with helicopter rides. Anyhow, I get out of this helicopter, and what did I find? This was back, you have to remember, in the 1960s, late 1960s. There were these guys with hair down to here, and all the cars were covered in flowers. So this was the, you know, I work on radicals. This was the era of the radicals in Berkeley. So that has been perfectly appropriate. So this weird looking guy, I mean, I'd never seen anything like this before. Took me to, I lived at International House till I met people and then got roommates. And so I went, that's where I went. And that was my introduction to Berkeley. And I think, in contrast to most people that go to graduate school in good places now, where the people you meet are your colleagues for the rest of your life, you know, they end up in academics or they end up in really good industrial jobs. That clearly wasn't true when I went to graduate school. Why wasn't that true? Not because it wasn't a good place, but because it was the Vietnam War days. So 90% of the people in graduate school were there to get out of the war. okay? Almost everybody. I was called a dirty red commie hippie. I mean, I used to leaflet against the war. Anyhow, one of my friends drank a quart of his own blood to fake a bleeding ulcer. That's the days. I mean, to get out of the war was why most people were in graduate school. So in contrast to the community of people you usually meet in graduate school and then you communicate with the whole rest of your life, there are very few people out of my era in graduate school that ended up being in academics. So anyhow, I went there. I was, everybody scared to death because I was away from home. Didn't know what the place was going to be like. And was trying to decide, in chemistry, you decide who you want to work with within a couple months of actually being in a place. And the first person I went to was this photochemist because I wanted to study the effect of light on painting. And that was my first rude awakening. I'd never really seen sexism before, but that was the first time I'd experienced the fact that I was different from men in terms of being a scientist. It never even crossed my mind before that experience. And then the second experience, there were two physical organic chemists, which I told you I was interested in from my undergraduate days. And one of these guys, I went and talked to him about his research. And the first thing he said was, well, since you're a woman, we will have to give you some kind of a project so that you can be a technician when you get out of graduate school. And so I basically, even in those days, I sort of didn't get it. I didn't have any confidence, but I stood up and walked out. And so, I mean, how did I decide who I wanted to work for in graduate school? There was a young assistant professor there named George Kenyon. This was back in the days when bio in chemistry was a dirty word. I mean, God forbid anybody that was a chemist should taint their hands doing anything biological. And George was one of the first hires at Berkeley that had a bio bent. He worked for Jack Buchanan here as a postdoc. Jack Buchanan's one of my heroes, by the way. And he also worked for Frank Westheimer, who recently died, who is the father of modern biological chemistry or chemical biology, whatever you want to call it. And I think just because I had this experience with these other two people, I didn't know anything about biological chemistry, right? I had no way of being able to evaluate any of that. Just because he was nice, that's why I joined his group. That was the basis for my decision in joining his group. And so that's where I was. Being in Berkeley was a very interesting experience. I mean, it was the days, not from a chemical point of view. In fact, I think I got a rotten education. It was interesting from a political point of view because back in those days, Ronnie Reagan was the governor of the state, and he used to do things like drop tear gas on campus, and then you would walk out of the building typically, and there would be a line of, standing there with their billy clubs raised to hinder any kind of protests. Or the chemistry building would have butyric acid put in its ventilation system. And so, man, that place smelled. You couldn't go in there for 2 weeks. Anyhow, being in Berkeley in those days was an interesting experience politically, but I would say, for example, I wanted to take a course with Dan Koshland, who recently passed away. This is not a good sign here, but he recently passed away. He taught a course with Jack Kirsch, both of whom were trained with Westheimer, and I couldn't get credit for the course in chemistry, even though the physical organic chemistry they taught was much better than the physical organic chemistry that was taught in the chemistry department at Berkeley at that time. You just didn't do things like that. So to see the shift now when I go back to Berkeley, I'm on the visiting committee at Berkeley now in chemistry and chemical engineering. It's really sort of remarkable to see how they've embraced the biological side of things. So really things have changed tremendously. And I think one of the things that had the strongest influence on probably the rest of my career, but at the time I didn't really think about it that way, was we had to give a seminar as graduate students. We had to pass our oral exams. That was one of the hoops. We had to give seminars. That was another hoop. And most people were giving seminars on the total synthesis of some natural product. And I was reading through the literature, and I read a couple of papers that really profoundly affected my whole change and thinking about science. And one of them was a paper by Tipper in Strominger in 1964. And that was a paper where Strominger did seminal contributions to understanding the cell wall of the bacteria, and that it was composed of alternating units of sugars, N-acetylglucosamine, N-acetylmuramic acid, and you had 5 amino acids and a peptide, and then you had a cross-links. And this paper showed how a conformation of penicillin looked just like dealanine at the end of this peptide, and the penicillin was a substrate analog of dealanine, and showed that binding in the active site, you could do chemistry. We understood enough in those days about those kinds of enzymes to irreversibly inactivate the enzyme. So that was, I think, my introduction to rational drug design and the power of chemistry to really think in detail about how enzymatic reactions and catalysts work, and then you could relate it to something practical out the other side. I will say that practicality has never been the focus of my research, but out of a lot of the basic stuff that we've done over the years, I think important practical things have come out that have been, the information that we've produced has helped people rationally design drugs. And that, I think, was reflected by this Tipper and Strominger paper I read in those days. And I think it's interesting because there was sort of like dead silence after I gave this seminar. Because this seminar was it just wasn't what people did. Nobody gave seminars on something this biological. And it's interesting because, 10 years later, I came back and people still remember this seminar. So this seminar had an effect on me. It also had an effect, I think, on people there about how exciting chemistry could be in relationship to biology. So having been at Berkeley for 2.5 or 3 years, I decided it was time to leave. I'd learned as much as I was going to learn because I didn't really think I was learning that much. So I told my advisor I was leaving, and he let me leave. And then I went and did a postdoc. I had no idea really what I wanted to do for a postdoc, but I thought I wanted to teach in a small liberal arts school. And so a bunch of people suggested, so in contrast to all of you who spent a lot of time reading and thinking and knowing the literature, I'd read and thought the literature, but I really didn't know what I wanted to do. I went to work for a guy named Jules Riebeck, who's now on the faculty at Scripps, who actually was an MIT colleague after I joined here. And I worked on the total synthesis of LSD, starting from tryptophan, okay? And he was at UCLA at the time, and while I was there, I met Bill Muma, who was on sabbatical from Williams College, and we had a really good interaction. He convinced me, even though they were looking for a physical chemist at Williams, that this would be my dream job. And so I wasn't in a postdoc in a very long period of time. I took the job at Williams College and had to teach physical chemistry and biochemistry, neither of which I knew anything about. Anyhow, so it was baptism by fire. I spent all my time studying, trying to stay ahead of the students. And that's where I got interested in biochemistry, was through that teaching experience. By teaching this, you really have to sort of know it. I had a good background in organic chemistry, which helped me focus on enzymes and cofactors and how they worked. And I got really excited about it. When I initially started, it was really pretty tough because there were no women faculty in the sciences at Williams at that stage, and the other biochemist was not easy to interact with because he would say to me, well, what about Amphenson's hypothesis? And I would know who Amphenson is. I said, I don't know who Amphenson is because I was learning biochemistry as I was going along. So it was pretty brutal, but what it did was give me enough self-confidence that I could take another step. And so that experience at Williams, by the time I was there for 3 or 4 years, I was starting to feel pretty good about myself and that maybe I could really do research at a place where I could work on my own ideas. And I was the first person at Williams to get an NIH grant. Didn't have a clue how to apply for an NIH grant, but applied for it and got it. I had 5 or 6 undergraduates work in my lab doing their senior honors thesis. They all published papers. And then I went to a Gordon conference and I met Chris Walsh and invited him to give a talk at Williams. So he was an assistant professor at MIT in those days. And he came in and I started talking. And he couldn't get a word in edgewise. I was so starved to talk to science to somebody that he was just like, let me out of here. You know, this lady is smothering me. But anyhow, he was really pretty instrumental in convincing me that I should get out of there and go back. And how do you get out of a place like Williams College to be able to do science in a place like MIT? It's not easy. So I applied for a fellowship and got a fellowship to work in Bob Abel's lab at Brandeis. And Bob Abels is, again, one of my heroes, not necessarily as a person, but as a scientist. I mean, he was just, he really was brilliant. And so that experience got me really excited about enzymes and enzyme mechanisms. And so it's been a long time till I got to that position. But I still was in my, I was about 27 or 28 when that happened. So because I didn't spend that much time getting my PhD, I hadn't sort of wasted that much time. What my experience allowed me to do was get enough self-confidence so I could take the next step. And after I went to Abel's lab, I never turned back. I mean, there was no way I was going back to Williams College. I really wanted to be in a place where I could start doing my own research and working on drug design, rational drug design. Now, nowadays, nobody does rational drug design. It's irrational drug design. But I still think there's a large place for rational drug design. And so when I applied for jobs coming out of Abel's lab, I looked at pharmacology departments. And my next job was as an assistant professor in pharmacology at Yale Medical School, which was a fantastic pharmacology department focused on cancer. Simon, get down. And so pardon me?
Which medical school?
Yale Medical School. So Al Sartorelli was the chairman of the department at that time. And so I got there. John Kosarich, I don't know if any of you know John Kosarich, but anyhow, he was hired at the same time I was. And we shared an office as big as that table. Okay, literally. I mean, I look at what young people have today, and I sit there and I say, you know, we were given nothing. Okay, our starting salary was like $15,000. Okay, we were given no setup funds, and we had an office this big. And so literally, when he would sit down and I would sit down, we both couldn't stand up at the same time. We were given a lab in a building away from the rest of the pharmacology department because there was no space, where the hoods didn't work very well, and I was doing a lot of synthesis with really not very nice molecules. And so that was also, I think, not particularly pleasant. But the beauty of a medical school was that I got to work in a lab and do my own thing. And so during that period is when I made a discovery, and that discovery was enough to allow me to get a job where I could have graduate students and take the next step. So at a medical school, you frequently work with technicians. It's not easy doing chemistry with technicians. And so I knew I would never have graduate students, and I got enough self-confidence when I discovered, you know what I discovered? This is sort of like amazing. How do you get tenure? Nowadays, there's no way you could get tenure doing this. I discovered that one enzyme could cleave a carbon-hydrogen bond. That's it. That was my discovery. But that discovery, you know, there are lots of people working on this, couldn't figure out what the hell was going on. And this enzyme happens to be pretty central to nucleic acid metabolism, and that's what got me immediate recognition for figuring that out. And I've been working on that system for the whole rest of my life, and I will work on it until I end up retiring. So anyhow, I made this discovery at Yale working in the lab. The thing is, I told you they gave us no money, but we had keys to all other faculty members' their labs, and nobody worked at night, nobody worked on the weekend. So John and I would just go take all of the stuff. You know, then we had stacks of stuff. They didn't even know if stuff was missing. Anyhow, all the equipment, all of the chemicals and stuff like that, we just went in and took it. And that's how we did our research when we started out. That's where we got all of our chemical reagents and stuff to carry out our research. So anyhow, once I made that discovery, I decided that I had a little more self-confidence and then took the next step. I applied yet again as an assistant professor. As Stuart said, he said it was 14 years. Shame on you, it was only 12 years. Anyhow, I mean, I guess, you know, so you can say, I mean, I watch young people now and how uptight they get about getting tenure, and I'm saying, that never crossed my mind. Tenure was never on my radar screen. My radar screen, I think this was from the way I was brought up, was just do the best you can and whatever happens, it ships. But I never was focused on a nature paper or a science paper. I just sort of watch what young people are focused on now, and it bothers me quite a bit. I was focused on solving problems. That's what I got excited about, solving some problem. And so I applied for a bunch of jobs, and I was lucky enough to get a job at the University of Wisconsin in Madison. And now, so I'd been in a chemistry department, and I moved to a pharmacology department. Now I moved to a biochemistry department. I would say this is interface chemistry. And at biochemistry in Madison was fantastic. It had a very rich history of making seminal discoveries on vitamins and how they work. Almost everybody that was anybody went through, including Gene, went through the University of Wisconsin in Madison. And so that was the first place that I was able to have graduate students in my lab, and still could work in the lab. I still worked in the lab full time because our teaching, again, in biochemistry was light compared to chemistry. So, I mean, to me, that was what I loved to do, was work in the lab. And so I was there for approximately 7 or 8 years, I can't even remember, and then again, the Chris Walsh connection, they invited me to apply for a job at MIT. I probably never would have taken the job here had it not been that I had aging parents and somebody needed to take care of them. I would have stayed at Madison. I think Madison was a great place to do science. But anyhow, I made the move for those reasons, not based on the science, and the rest is history. I moved here in 1987. I think what's sort of amazing is that even in Madison, I was the first tenured woman in a biochemistry department in 1984, pretty bad. And here, I was the first tenured woman in chemistry in 1987. Not good. Now, I think I take a look at people all over the place now in chemistry and biology, and I see a lot more women in the pipeline, and I think things are starting to shift. So I think the great thing about MIT for me has been all the smart people I interact with. I think it's been allowed me to do science I never would have done if I hadn't moved here. I've had great collaborations with 3 or 4 labs on campus that really, through mostly interaction with their postdocs, I really have learned an incredible amount, and I think they've also learned a lot about biochemistry by working with my lab. So, I mean, that's what I have to say, and I'm happy to answer any questions. What am I going to do when I retire? I am going to garden, and I'm going to hike, and I'm going to read books. Anyhow.
Why are you even thinking of retiring?
Oh, yeah, it's an interesting question. I think you need to know when it's time to hang it up. So I'm not ready yet, but I'm thinking about it. What I see is, I think this is, yeah. I think what I see is, and this happens to every generation. You see how science has changed. Science has changed tremendously. Where you were studying enzymes and how they work, and I mean now we're into whole cells and whatever systems biology is. I mean, and the methods we use. So you keep learning and you keep changing, and I have. Otherwise, you wouldn't survive if you didn't keep retraining yourself. But the way people view science now is very, very different from the way I do science. And so I think you need to know when it's time to step aside to let the young people with all the crazy ideas that you think don't have a bat's chance in hell of working, get in there and make the next set of important discoveries. So yeah, I think I'm thinking about retiring for those reasons. I certainly wouldn't quit science. I like science too much. But, I might go someplace else and teach or work. I mean, one of the things that I've been thinking about doing is taking courses in energy and climate, and trying to work at that level to educate lay people about the importance of these problems. And one of the other things I think that nowadays we have a National Institutes of Health. We've had it for many years. I think we should maybe start thinking about cutting back the money at the National Institutes of Health and starting a National Institute of Energy and Environment, because we won't need a National Institute of Health unless we start paying attention to problems like that. So I picture myself not sort of quitting science, but extracting myself from this place and contributing in other ways after I decide to leave this place. So.
You enjoy teaching?
Yeah, I love teaching. I'm not sure what the students think, but I love teaching. So why do I love teaching? I think it's for selfish reasons. I love teaching because I like to learn stuff. And so most of the things that I teach are not introductory courses because I'm not charismatic enough. But I teach sort of advanced courses. And any advanced course you teach, you can say, oh, I can go to the library and read these 500 papers on translation or transcription or whatever. And what that does is it allows me to learn stuff and it allows me to think more broadly about my research ideas. And I love what I teach. And so I think the way people learn is on their own, not by the teacher. The teacher can only inspire. And if people can't see my enthusiasm, that's their problem. Okay, I mean, I think I wear my enthusiasm when I teach on my sleeve. And if that doesn't do it for them, then you know, I don't know what. But I love teaching for that reason. You know, I've been teaching a course, the equivalent of 775, sort of 508 macromolecular machines, and I love teaching that course. Yeah. Anybody have any questions, comments? Don't be intimidated.
Did you
I don't
teach high
bite.
school?
Huh?
Did you teach high school?
I did teach high school. When I first, back in those days, they really needed people to teach high school. My mother was a high school teacher, so this was when I was also in college. So when I would finish at Penn, which was in early May, then there would be May and June, and they would need substitute teachers. So I decided that's the way I would see if I could do this. And it was horrible. I mean, I had a kid pull a knife on me, and you know, I turn my head, and I get 50 spitballs in the back of the head. I had no control over the class, no matter whether I was nice or whether I was not nice. And so that experience, actually, for 2 years, convinced me that there's no way that I would ever be able to control the class, and I wouldn't be able to teach. What I'd be doing is discipline duty. Which, you know, I think that's why I guess teachers have to take all these courses to figure out how to control a few people in the class so you can actually get people excited about some kind of science. So.
Are you teaching chemistry then?
I was teaching all kinds of, so it was anywhere from the 9th through the 12th grade, so I was teaching math, physics, chemistry. It didn't matter what I was teaching, even when you had kids that were really, I mean, I even know some of these kids now, it's sort of because, you know, it's from Worcester, but, you know, they remember throwing spitballs at me. This is a good class, you know. So, I mean, it was just, I just didn't have any, I had no control. And so, you know, the high school thing, I mean, it could be that I could go back and do that now. I don't know. I would have to figure out how to get control, though. But I would have to take a few education courses to figure out how to control.
Your father was a professor.
Right, he was a mathematician.
Did that have any influence on you and your decisions to how to?
No, I think both of my parents, you know, came from immigrant parents that had no education, and they both worked very hard to educate themselves. They both were educated somehow. In the family, it was ingrained on all of us. I had a sister and 2 brothers, how important education was. We lived in a really tiny house. And so that was the other thing, you know, concentrate, I can concentrate. You know, I can be sitting there, the whole house can be caving in, and I don't even know anything's happening, because when you're in a tiny little space, you learn how to concentrate. So I think that experience really told me how important learning was. And also, I think, for both of my parents, that you could do all you could do is the best you could, and that was good. That was what you did. And if it wasn't, you know, you might not do as much as you wanted to do, but if you did the best you could, that was good enough. And so I think that has always stayed with me. Yeah, I think my father's most important influence on me had nothing to do with mathematics, but I used to play bridge. I started playing tournament bridge when I was 9 years old with my father, who was a life master, who was a really good bridge player. He loved basketball. My passion was the Celtics. Man, I used to sneak the radio to bed. Nobody knows who Johnny Most is, but I used to sneak this with Bob Cousy and Tommy Heinsohn. Man, they were such good. And I used to babysit for Tommy Heinsohn's kids and Bob Cousy's kids. They lived in the neighborhood in Worcester. And so we used to get free tickets to the Celtics game. So my father brought that passion to me. And the other passion he brought to me was tennis. He was a really, really good tennis player. And so he used to take me, back in those days, there was clay court tennis, to Longwood Cricket Club in Boston to watch the tennis matches. So I think that was probably his most important influence on me. Was I interested in math? Yeah, I mean, everybody in my family was good in math. It was probably genetic in some way. But I knew that I was never going to be a mathematician. I just wasn't, Being a mathematician means you're walking around in 6 or 7 dimensional space, and you really don't know where you are. I wasn't in that. I was not in that league. So science, whatever. I mean, I never ever doubted that I couldn't work on whatever I wanted to work on growing up as a kid. It never occurred to me I couldn't do something because I was female, for example. That just didn't. I was going to do what I wanted to do, and I would do the best I could. So.
Do you feel like you had to make any sacrifice to get to where you are?
Well, I mean, I guess it depends on what you call sacrifice. I'm doing what I love, okay, most of the time. So how many people spend their whole life doing their hobby and get paid for it? Okay, so you can say, I'm not married, I don't have any kids. I have my sister's kids and my brother's kids. I have a dog. I'm not sure, given how much work the dog is, I'm not sure what I would do with a kid. Anyhow, so yeah, I mean, I got to do science, which I really sort of loved to do. So I never look back. So, yeah.
Through the talk, you keep on talking about the extra things in your life that kind of have affected your science, like your interest in art and your interest in history. That has kind of, you mentioned people that have contributed to science over the years. How do you think that lack of art and history teaching in schools is affecting how people look at science, particularly the new generation coming through?
I don't have a clue what they do in schools anymore. I mean, they don't teach anything like that anymore. They don't teach kids how to read and write anymore. Okay, I mean, I kid you not. I mean, I would say, this is a terrible thing to say because there are my graduate students in the audience, but I mean, I would say I spend more time teaching people what a sentence is than teaching them how to do science. I mean, people don't read anymore. They watch the computer. I mean, this is a generation saying it's time for me to retire. They watch the computer or they play computer games. I mean, that's beyond me. And they don't, so where do you learn how to write is probably from reading a lot. People, I don't think, do that much anymore. And they don't do that much writing anymore. So, and so the question is, what will the future be? Maybe we won't have reading and writing anymore. Everything will be done on the computer. I don't know. I mean, so I think I'm not going to judge what they should be teaching in high school anymore. Just like I'm not going to judge who the people are we should be hiring in chemistry. I think that's left to the next generation. They think about things very differently from the way I think about things. And that's what happens to every generation when you get into this age bracket. Things change. If it doesn't, it wouldn't be exciting. I mean, science, look at what's happened in science. I think I probably lived through the best. It's just totally amazing what you can do. I used to spend 3.5 months in the cold room isolating a microgram of my protein. Okay, a microgram. My students, in one day, isolate 2 or 3 grams of protein, because of the revolution in molecular biology. We have the genome. You can go there, you can find new pathways by looking at organization of bacterial genomes. You can do, you can put unnatural amino acids into proteins anywhere you want to study the details of how they work. It's just totally mind-boggling what you can do nowadays. And it's just the tip of the iceberg. Things are changing very, very rapidly. So I don't want to second guess where high school's going or where college should be going. I think it's something that we need to discuss. How should we be teaching? I think that's an important issue. We've been doing the same thing teaching for for decades. We had the same tenure decision for ways to decide tenure for decades. You know, maybe it's time to do some thinking and some changing. But I'm not the one that's going to make those decisions. I think the next generation needs to do that.
JoAnne, the discovery idea must have been a real aha moment, right?
It was a what?
Aha moment.
Yeah, it was. I mean, I can remember this day. I remember this till the day I die. I was so excited. This is the thing that also made me leave Yale. Anyhow, I was working on this mechanism-based inhibitor. Okay, so that means that if you understand the mechanism, it can enact the enzyme, catalyzes self-inactivation. And I had gone over to chemistry. I had isolated some of the products. We work on an enzyme where the whole substrate blows apart into 15 pieces. Anyhow, I had taken an NMR spectrum in chemistry and came back over, and I was looking at the spectrum, and it was inorganic pyrophosphate. And then the light bulbs went on, and I understood the whole mechanism of how it worked. It was on a Saturday morning. I remember I went running down the hall. There was nobody there. I went into the chairman of my department, Sartorelli's, and I was like jumping off the wall because I was so excited about this discovery, because all of a sudden everything came into place. And he started talking about something completely different. And I said to myself, oh my God, I got to get out of here. That was it. I need to be in a place where somebody can appreciate why what I'm doing is exciting, so I can share some enthusiasm with people of like mind. Anyhow, so yeah, it was.
Have you many such aha moments since then?
Well, I think when Stuart was in the lab and when we saw an exchange-coupled thiol radical, that was pretty exciting.
That was exciting.
Yeah.
You were there in the EPR.
It's
room
probably
in the
waiting for the
Yeah, no, I remember
spectrum to
that.
come on. It's probably true. So, you know, everybody he says that's weird. But see, the thrill of discovery, there is no replacement for doing it yourself. So at least if you're there, and then, you know, I mean, you're participating in the thrill of discovery. Yeah. I don't know.
JoAnne,
Yeah.
have you been reasonably satisfied with your career here at MIT?
I think MIT is a tough place. I'll leave it at that. I think MIT is a tough place.
Elaborate about what you
I don't
mean?
think so. No, I don't think so. I think I focused on the good part. You know, the part where I have had outstanding collaborative interactions, and I've had good students in my lab. I think that's the pluses of it. But I think, yeah. I think it's not a collegial place, and I think it's not a compassionate place. Leave it at that. And so I think different people have different experiences. That's just my experience.
It struck me in your presentation that you have something else in common with Chris Walsh, who both did postdocs at Bob Abrams.
Great. Well, I've been friends with him since 1971. And, you know, over the years, I've been on almost all the students' thesis committees. And so he has been my lifeline to enzymology, really, because, you know, I talk science to him, and we talk in detail about science. I don't see him as much as I used to see him, but a lot. His daughter, for example, worked in my lab for a summer. So, yeah, I mean, he hired me, and then within a microsecond after he hired me here, he left. I don't know if that's a comment on something, but he moved to Harvard Medical School. But over the years, I've been friends with him and his wife for a very long time. So that has been a really positive interaction. Yeah. Yeah.
JoAnne, what advice would you have for the younger scientists, students or postdocs?
What do you mean advice? Advice about what?
In terms of doing science and choosing your career.
In choosing your career, I think everybody has to do their own thing. I think the key to success is motivation. I think it doesn't matter what. I don't think it matters how. There are lots of things that go into making somebody successful. And nowadays you have to wear so many hats, it's sort of unbelievable. And nobody wears all those hats well. But I think you making the decision, you've got to decide what you're passionate about. If you're lucky enough to get a job where you find something you're passionate about, then I think you've been successful. I think most people don't do something they're passionate about. How you find that, I don't know. I mean, you've got to find that as an individual. And it doesn't have to be academics where you pursue your own thing. I mean, there's a lot of downsides to academics. You spend, when funding is bad, you spend all your time writing grant proposals. It's stressful. But, and there's a lot of good sides to industry, which people used to dump on when I was in graduate school, because you can be involved if you're willing not to have your name in the lights, which, I don't care about at all. You could be part of a team to make discoveries that could lead to some new drug. So that could be exciting too. And I think if you're in a good company, you could participate in everything if you have enough energy. I think most people don't. I think they're myopic and focused on their own little area. But I think if you had energy and wanted to work on the bigger picture, I had one student at Merck who did that, and now has a pretty high up position because she was interested in the big picture. So I think it's an individual choice. I think you got to find what you're passionate about. It might be bench science, it might not. It might be communication to the rest of the world about science. It might be, using your scientific background to teach. It could be using your scientific background to learn more about energy and figuring out how to communicate that to the rest of the world. I think it's an individual thing. And then, the balance of what your life is like, that's an individual decision too. And everybody needs to make their own choices about that. So, yeah.
You just said in MIT, first food policies, which is interaction and discussion.
The what? I can't hear.
Interaction. You said that MIT is a good place to interact and discuss with students.
Yeah.
But on the other side, you said this is a place not with competition. So how do you deal with that part?
How do I deal with it? I ignore it. I go into the lab and focus on my research. When something bothers me about lack of collegiality, I sweep it under the table and go focus on something I'm excited about. That's the way I've done everything all my life. If something is really upsetting me, I just go read somebody's paper, like, Steitz's X-ray structure of the ribosome. I was on a high for a month after I read that paper. So I just bury myself away from the parts that I don't like.
Yeah. So outstanding you are, so you don't have to tolerate, right?
I don't have to tolerate it? I don't know. I just focus on the good most of the time. Most of the time I just focus on the good. Everybody has good points and bad points. And so if you focus on the good and ignore the bad, you'll be more happy. Even dogs. Okay. You a good doggy, Simon. Okay. Anybody else? Yeah.
So you're consulting for big companies, I suppose?
No.
No?
No.
Are you on the board of any company?
No.
Never was?
No. I think that that might be different for the younger generation. For my generation, I think that's not unusual. So. All right. It's nobody else. Okay.
Thank you.
Thank